Hello, my name is Martin Salguero, and I will represent the University of St. Thomas in Houston, and I will be presenting our project, which is the characterization of viral communities carried by male and female Aedes mosquitoes collected in Houston. Our project focuses on characterizing the viral communities carried by different species of mosquitoes in Houston. While we know that mosquitoes carry important human viruses, like West Nile and dengue, the true collection of viruses that they carry is not well documented. Viral ecology in general is only now beginning to be characterized thanks to the sequencing techniques that sidestep the need for culturing. In our research, we are using metagenomic approaches to sequence the total genomic sample of mosquitoes and using a bioinformatics workflow developed in-house to identify viral sequences present in our samples. We are specifically looking at different species of mosquitoes commonly found in Houston, such as species from Aedes, Culex, and Anopheles genuses, and comparing the viral communities found in males and females of those same species. This work is relevant because Houston is an ideal breeding ground for mosquito populations due to a constant rain and accumulating pools of water the city experiences every year, and because they do carry serious diseases like previously mentioned, such as West Nile, which have been previously documented in our city. But we are interested in characterizing as many viral strains as possible in our samples and comparing the viral collections between sexes and across species. Our project does involve a little bit of field biology, such as capturing mosquitoes using traps, molecular benchwork, which includes nucleic acid isolation, and computational biology, which has to do with sequence data analysis, in order to reach our goal of characterizing the viral communities in these organisms. So moving into our methodology, our traps were set out at strategic points in the city, at first to coincide with areas where West Nile positive mosquitoes have been found, as seen in Figure 1, and then to other areas in the city to increase our data pool. Then these captured mosquitoes are brought into the lab where they are frozen until they can be processed at a later time. So once we have these mosquitoes in our lab, we separate our captured mosquitoes using morphological characteristics such as abdomen shape and coloration, striping patterns, and antenna and mouth parts. And we use that criteria to separate individuals and sort them by species and sex. So as we see in Figure 3, there is an example of how we differentiate between male and female mosquitoes, which is by their distinguishing antennae. So this antennae distinguishes between males and females by either being fuzzy or non-fuzzy, fuzzy for males and non-fuzzy for females. So as we can see in Figure 4, individual mosquitoes that have been separated by species and sex are then pooled into 10 milligram sets for nucleic acid extraction. We then grind these mosquitoes up to homogenize them and use protocol kits to isolate their total DNA and RNA. So a part of the DNA that we extract, we use to verify the species by using PCR to amplify the cytochrome oxidase CoA1 gene. So once we have that, we send off our samples for sequencing to a private sequencing company and then use BLAST to confirm the genus and species of our samples. A sample of those sequences can be seen in Figure 6 and a gel electrophoresis that we create a lot in order to confirm the presence of those CoA1 gene sequences in that sample. On the other hand, RNA is set aside and used to make cDNA or complementary DNA to make a library of cDNA by RT-PCR or reverse transcriptase PCR. So we can sequence and transform the RNA into cDNA, which allows us to sequence and analyze the RNA viruses that may be present without having to work with an unstable and easily degradable sample of RNA. So once that's done, the total DNA and cDNA are pooled and sent off for sequencing using 454 technology. So far, we have completed one complete round of these experiments with male and female samples of Aedes mosquitoes, and when we received our sequences, our metagenomic sequence data needed to be sorted and curated in an automated way because of the sheer size of this data. This is why we devised a workflow in-house, as we see in Figure 5, that automatically converted 454 files to FAFSA files, and then automatically blasted each individual sequences in bulk. These results were then filtered for viral sequences hits, and all unknown sequences were separated into a new file for further analysis as well. So, our initial results, as seen in Figure 7, show that we can sequence both DNA and RNA viruses. We were able to identify 51 viral families in our female sample and 40 viral families in our male sample. Interestingly, there is a marked difference in the abundance of viral types between the two sexes. We found a large representation of single-stranded RNA viruses in females compared to males, and males had way more DNA viruses. While differences between the sexes were expected given their different feeding habits, we need to do more sequencing experiments to determine if these differences are biologically relevant or a consequence of sequence biases in our experiments. As an initial proof of concept, our experiment shows that we can produce sequence-grade DNA and cDNA samples which we can use to identify both DNA and RNA viral sequences in our samples. We hope to continue to sequence our other samples and to determine if the viral distribution patterns are sex and species specific. These results will help us to better understand the true viral collections carried by mosquitoes in the Houston area.